So, good morning, everybody. Time for second lecture in quantitative big imaging. And today we are going to talk about ground truth, building and augmenting data sets. And well, let's see, I need to switch that one. No, it should work, yeah. So, what we are going to talk about today is first creating data sets. So there are a lot of famous data sets around. There are different types of data sets. And we're also going to look a bit what is a good data set. And of course, building your own, how to do it, which methods you can use for it. And then on top of that, we are going to also look into augmentation. That is when you have too little data, then you can try to modify your data to kind of expand it. And you can also see a little bit about the limits this gives. Finally, it's not actually about data, but more about algorithms. We're going to look at some ideas behind having a baseline algorithm for your evaluation. And then the usual modules that need to be loaded. And there are some reading here, actually a lot of reading. And it's an overview of different topics related to how to use other people's data. And this goes on, also creating data sets, crowdsourcing. There are also two links to different augmentation tools, how you can actually a little bit more interactively create your augmented data. So why other people's data? You have your own data. You want to analyze your data. So what is the point in looking into other people's data? And in principle, let's see first what other people say, people who have been working a lot within data analysis in different ways. And first we look here at a guy called John Taylor, and he is at Facebook, and he says in principle he has learned more from a few well-known data sets than from all his fantastic machine learning algorithms, because using yes,
I think we don't see the screen share of the presentation.
You don't see the shares? Oh, that's not
I mean,
nice.
I see one bar, but I don't think it's your presentation.
No. Okay, good. I'll check about that. And let's do a full screen sharing. Then share, and now you see it, I hope.
Yeah, looks good. Thanks.
Good. Perfect. So this is one view of it, that you really need good data to get new knowledge. Otherwise, you have a bunch of data, you have a bunch of algorithms, but it doesn't come really good results out. Another view of it is comparing between how many lost hours of sleep you have when you compare a PhD who is looking at his data, or someone at Tesla. And it's a little bit small here, but the red bar here, or the red little slice here, is about data for the PhD. And then everything else is about algorithms and stuff that to analyze the data. At Tesla, it's not perfectly the other side around, but they spend a lot of time about creating, collecting, and making sure they have the right data, and then some time on working with the algorithms and modeling. So it's a different world out there. Of course, you have your little data that you can capture, and then you have to do something with it. At Tesla, they have it completely different. They have a full staff of people just doing that. Another view of it is got a couple of tweets from Catherine Scott, and she considers her work as 95% is about making the data in a good way that she can work with it. And then afterwards, she lets some algorithms for 5% of her time doing the machine learning. So data is important, and someone has claimed it is the new oil. So some kind of what's called snake oil or whatever. So there's a lot of bad data out there, and people are selling bad data for people to do machine learning, and that's not really a good thing. And to test your algorithms, you need really good data. You need data where you know what you have. And if you don't know what to expect, so how can you know that your algorithm is actually doing what it's supposed to? And if you have a lot of edge cases, how can you make sure that it actually works for all of those? It's about data balance, that mostly things happen in the middle, but there are some very interesting spots on the edges, and those are actually the ones you want to have, and you want to be sure that you really get those. And in some cases, there are even algorithms, like for Facebook, apparently they develop new algorithms every hour. And how can you actually know that algorithm A is better than algorithm C or whatever? And for that, you also need a lot of data to really test that the algorithms are performing in the way you expect them to. And also for machine learning, which we will talk about, but not only, it's really important that you tell the algorithm what it should do, because it only learns what you gives it, sorry. So the simple example in biology is counting cells. You have an image, you have the cells, that's quite easy. But if you want to look at the difference within different cells, is this cell healthy? Is this bad? And when you're going to look at those gradual changes, then you really first needs good expert knowledge to mark up the cells that really contain the information that you want to tell it. And that's not very easy, and it's a very time-demanding task to find it. Sometimes it's actually possible to do it with traditional analytical methods, but very often in image processing, the eye is very smart, but the computer is not so smart. So you really have to tell it what to do. And that is done by having good data. Then one good thing is, once you have used the data for your project, it's not consumed. You can actually reuse it for another project or someone else's project. And the way to do it is that you really need to have to mark up and label, document what is the data, what were the conditions, and that needs to follow with each data point sometimes in order to make it available to other people. And then the other side is that usually when you start building your own algorithms for doing the analysis, by nature, it turns very messy, turns complicated. It was done under stress, and you find a debug here and debugging here, but you didn't think much about the structuring. And in particular, if you want to finish up in time for some project, then the quality of the code is usually suffering. But if you have at least good data that you can compare with, so you know what it's supposed to, then at least you have better chances to get things right. So it saves time. Well, it has first a very boring step of preparing the data, but in the end, it actually saves time to have good marked up data. Of course, a lot of people have worked with data sets. I think the most well-known one is the ones with dogs and cats, telling if the dog or if the cat, or I think a cupcake. There is a nice example of where you have a little chihuahua comparing with. Is there a question? Okay. Other data sets are about handwritten digits. That's the MNIST dataset. And I'm going to show a little bit more about these. There are also a lot of medical data sets for diagnosis of different diseases. A lot goes towards cancer detection. And these data sets can be used as a starting point for other data or for other analysis. Sometimes it doesn't even have to be what you are looking for, but it has the same behavior. I think I'm not sure if I show it, but anyway, I had a student who wanted to find root structures in soil, and he trained part of his machine learning algorithms using retinal scan blood vessels because they have about the same structures, and from that he could pre-train using that because there is a lot of data very nicely marked up, and then with the little data he had, he could actually boost his own algorithm. So here we have the MNIST data set, and yeah, more or less nicely written digits, 0 to 9, and each is labeled. It's also prepared in a beautiful more or less black and white style, so it's very easy to detect. Yeah, you don't have to do a lot of pre-processing before. Of course, you can make them worse, but at least here it is quite nicely prepared. Then we have ImageNet. It's a huge database, and it's categorized in a lot of different categories. It's very small text here, but you can say, oops, no, that was not what I meant to. Where is my pointer? Yeah, here. You can say first entity, it's an object or substance or matter. Then you can see if it's organism, artifact, etc., and it's categorized down into these groups. And then you can say you want to work only with plants and then detect if it's a flowering plant or not, for example. So then you can just extract that part from the data set. And then you have a lot of different architecture for machine learning, and you can see the complexity is increasing with the amount of computational power and also the performance. And what was now the parameter here? I think the error rate is decreasing with the number of layers. And here you can see on some ImageNet tests, classification tests, you only have 4% error in this. And in the beginning, when they started, they had 28% error. So it has been a lot of improvement with available computational power. Then we had the BRATS as segmenting tumors in MRI images. And here someone has already marked up a lot of brains with tumors and probably also without tumors, I hope, because you have to have the ground truth also when there is healthy patients. And here you can then see what it can look like. Different color probably represent different types of tissue and different degrees of disease. So what can you see from this? There are a lot of data sets, and there are also different ways how to handle the data. On some way, you can have handcrafted features. Someone is actually sitting down, marking up the images by hand. And in many fields of science, you can do this. You have always someone who knows how to mark up the data. If it's not yourself, some of your colleagues or collaborators know how to mark up. It is very time consuming, and you can have people looking at it. You can have handwriting experts for topology of the graph. You can have people to actually identify people in the ImageNet, or you can look for health in the tumorous tissue, for example. If you look at the machine learning approach, someone who works with machine learning doesn't have to know the topic that they are actually working with. They can be computer scientists, and they are looking for anomalies in the climate. They don't know anything about the climate, but still they can find the anomalies. Because these machine learning algorithms, they can actually find the patterns if they have the right data. And about working with these large data sets that are available out there, they can actually tune algorithms to be better and better and better. There are even like teenage boys sitting in their basement building up some funny algorithms, and they perform quite well, actually. Probably don't know exactly what they're doing, but they put stuff together and get good results. But the problem is also, if you want to work with someone else's data, you have some drawbacks. And Kasi Kosilot, I can never pronounce her name, Kosilkov, she gives a lot of good lectures. Here is one a comment from a paper she wrote on how to work with other people's data. And her first analog is to say that using inherited data sets, it's like inheriting a toothbrush from someone else. It's an act of desperation. You can do it, but it's not the ideal thing. And you have to do it sometimes, just because collecting data is really a luxury. You maybe don't have the funds. You may not have the lab times to do it at all. So that's why you have to actually get this in. And it's very easy, or comparably easy, to get someone else's data, but it's harder to trust. You really have to be sure that the one who prepared the data actually did the right thing. In this paper that I link here, she lists afterward how to deal with this data. And also an important conclusion is also that you have to be very honest when you say, I have used this and this data, and that's why we have maybe some more uncertainties when working with it. Another thing about sharing data is something that is been very popular or important the recent years, is the FAIR principle. And nowadays, every funding agency, they require you to actually publish your data openly once you have at least published your own publication. So usually you have some kind of safe time to prepare and show your research, but after that, you should always publish your data. And the FAIR principle is built on the concepts findable. So it needs to be in a database where you can at least find it. The next one is accessible. And with that, one thing is there should actually be somewhere where you can download it, ideally. And that is often done using DOIs, as a document object identifiers. And they are what's it called? Every DOI is just pointing to one object, so there is no confusion about what it is. Interoperable is something about how available, how you can actually use the other one's data. So if you use some of your obscure data format that no one knows, then no one can use it, actually. And that's what the interoperability is about, that you can take data from one place and bring it in to somewhere else. Reusable is, again, that you need to add sufficient metadata on your data set so that it's possible to understand what's actually going on. And the whole FAIR principle is discussed and presented in this Wilkinson et al. Paper. And within the neutron and photon science, there is now the large initiative called PANOSC, which is a European, I think it's only European, collaboration to make data sets from neutron and photon sources available to the public. Then there are, of course, a lot of other data sets available. There are a lot of Grand Challenge. There is this Grand Challenge org, which has a lot of challenges in the biomedical area. Kaggle is kind of a competition data portal where you can get a task and try to solve it best way. And sometimes you can even win some money if you're really good. Can be companies that put up a question, solve this for us, and then the winner gets the money. With Google Dataset Search, you can also look up data, actually quite wild collection of data. And also in this Wikipedia page here, there is a list of different databases categorized into different topics that where there are available databases. So what is now a good data set? I've been saying all the time, good data set, good data set. First of all, mostly a good data set is large. Many of these methods, when you want to work with them, you need a lot of data to make sure that you're actually doing the right thing. You need the data, amounts of data to get the statistics. The other thing is diversity. You need to have data that represent all things that can happen within the data, and in particular, the things you want to detect and identify. Finally, very important, annotations. You have to tell this image contains this and that, or these regions correspond to this and that. That is very important because otherwise it's just data. No one knows what it actually is. So, in principle, small data sets, of course, take what you have. But if you can get more data, it's usually better. And if you have really complicated problems, then you need to really make sure that the data reflects this in a good way. And it could be also that you're looking for very, very subtle changes within the grade levels that says, this is good, this is bad. Another thing is also class imbalance, coming back to the root problem that I had mentioned before. If you have a pot filled with soil and there's a little plant growing with this root network, the root volume to soil volume ratio is probably 1 to 1,000. And that gives us quite a big unbalance in the data. And that also needs to be handled in a good way. Otherwise, you will more or less have a classifier that says everything is soil. Fine, we're happy, but not. So what does the data really look like? We have lots of different types of data producers. So we have different scan types, different imaging modalities. We have reconstruction algorithms that does different things. Depending on the experiment conditions, we have different noise levels, illumination levels. Sometimes rotation can be disturbing, but you have them, or distortions from the optics. Can also be variations in colors if you have color data. And you also need many examples, as I said before. So one example here of male breast cancer, it's very rare, but it exists. So if you only have one case of that, it's not really representable for disease as such. You only have a single observation. And then it's very hard to make generalizations based on that information. Then, of course, meaningful labels. As before, I just said labeled data. So they should be very clear on what they label. The worst thing is if you say something very fussy, it's kind of healthy-ish. That's not a good label. So it must be unambiguous so that different people can actually identify this is actually what you're saying it is. And even better, and it's also one way that it can be, it should be possible to derive it from the images themselves. So the image itself must be able to tell what it is. For example, as this example here, if you have an image of a lung and you have the label that this person couldn't pay his insurance, it's not telling much about the image. You can't do anything with that. Or else you have to go to a case where maybe 4 people usually get this disease. That would maybe be a connection, but it's really a bad one. It's not really working. Very important, it must be quantitative in some sense. You should be able to count or tell something from the image. And it shouldn't be too obvious. So, I mean, if you say this image is bright, you can actually measure it very easily. Or you can say that this object is small. Yeah, sure, it's small. You can measure it. You can measure the area. So you don't have to give that kind of labels unless you have a very, very good reason. Then what do you need these different data sets for? So there are typical topics. We have classification, regression, segmentation, detection. Can even be that you are working with filter methods and want to check how good a filtering technique works. Then, of course, you don't need so much of labels, but maybe you need information about singular to noise ratio to categorize them easily. So classification is taking an image, looking at it, saying, this is a dog, this is a cat, and that is the task of the classification. It just gives you a global label on an image. And it should be exactly one category. It couldn't be cat, dog. They don't exist. So if it's between, then it's not good. So you need to really be distinct about what it is. They don't have to be ordered in some sense. So you don't have to have increasing importance or something like that. It's just saying, this is this, this is that. A little classification example that I'm going to run is we have here, oops, let's see if I can bring it up. Can't. Yeah, anyway, this is the MNIST dataset where we have the handwritten numbers, and we also have the labels associated with them. There are actually cases in this database that are very ambiguous. Is it a 1, is it a 7, or sometimes even is it a 9 or is it a 4, as you can probably see soon. Then we have regression, where you actually put the labels, connect them with a value. So for example, if you take a picture of a house and you train your algorithm to tell the value of a house that looks like this, or looking at a hurricane or satellite image and telling, is there a risk for hurricanes forming? Here's another regression example where you can read more about if you are interested on this link here. And it's about a lot of X-ray images of children's hands. And from the hand shape, it should be possible to tell the age of this child. Then we have the segmentation task, where you taking one image and you start looking at details in the image. So in this case, you want to look at regions. That example I have here, you look at regions that correspond to cats and regions that correspond to dogs. This is a quite fantastic image that they managed to have so many cats and dogs sitting still on the same time, but that's something else. And every pixel will be assigned a label. In this case, we have cats and dogs, and actually also background that we would like to see. And the questions that you look for, have in this case, where are something? So like, where are tumors in the image of a lung? Identify the streets in a satellite image, or of course, the cats and dogs, as I show here. Then going to a more scientific question, a different scientific example, where you have looking for cell nuclei in a microscope image. You have the image, which is kind of color-coded from some staining, I believe. And then afterwards, you want to tell where are these cells. And well, now it's a very dark example, but on your own screen or at home, you can look at and see that's actually each cell has got a label. Detection is to look at an image and predict where and which type of objects appear. Could even be very simple. So if you have a car driving around in a movie, it's enough just to tell you have the car within this box and it's moving. You don't have to tell the exact pixels where they are, but more telling that things are moving around and you can identify them. And it's even possible that the things overlap. So if you have a street view where you see two cars coming, you should still be able to see that which car is coming. So not that it comes like this, they overlap, and then it looks like that car is driving back and that car is driving back. That's not the purpose. So you have to be sure that you have identified them clearly and can follow them in the right way. Here you have a detection example from medicine again. So you have the lungs, and we are looking here at opaque regions in the lungs to detect some disease. And this is also a thing that you can see on Kaggle. So if you want to play with this data set, you can actually go follow the link to Kaggle here. Other possibilities, well, there's a lot of different possibilities in looking at why you need the data, which kind of data you can work with. There is one example here, making horses into zebras if you want. So there is some algorithm that actually converts a horse picture into a zebra picture. Could also be used for image enhancement, as I mentioned before, that you want to actually learn some algorithm to enhance the data somehow, and or going towards super resolution. That is when you make several images, actually usually a little bit offset, maybe randomly, and then from that you can actually resample the image and achieve higher resolution than the imaging system actually can give you. So now we are come to building your own data set. Until now was mostly about having other people's set. Building your own data set is very time consuming. And first thing is you have your experiment with all the planning, etc. That is one thing. But afterwards, once you have the data, say you have a gigabyte, a terabyte of images that you need to mark up somehow. That takes a lot of time, much more time than the experiment itself. And unfortunately, there is not so many good tools out there. And also the markup or the labeling is very problem specific. So it's really targeted on your problem. Sometimes maybe you can do something that can be recycled, but or you can do with some easy moves, you can make it recyclable for other purposes. Of course, everyone can recycle if they like to use your kind of investigation, but it's not always that you want to do the same investigation on that data. So there are code-free ways of labeling, marking up the data. So very easy is to just put images of different types into different folders. Then you have already done some kind of database work. You can do tables where you have an Excel file where you just list image name and then some information that you want to tell about this image. Then you can work in different drawing, painting programs to actually draw the regions that you want to mark up. And by own experience, this is really, really time consuming. Then there are software tools for data labeling. Each one of these are links, so you can really go and see what you can use. So there are different tools for classification, for segmentation, object detection, and other tools. Then there are, of course, also a couple of commercial tools. So there's a lot of ways to do this data labeling. And here is one example. Again, thanks to the light, the contrast is not very good. So hopefully on your own screen, you can see it. This is a neutron radiograph. And in neutron imaging, we always get these bright spots, outliers, as a consequence from the measurement technique. Neutrons captured produce gammas, and these gammas, they hit the detector, and then we get this white spot. We want to get rid of them, and different ways of doing it are available, so through filtering. And in this case, I want to do a demonstration how to detect these spots using machine learning. So I was sitting down in a 2000 2K by 2K image. It took me 8 hours to mark up all these dots in one image. So this is not really a feasible way of working, but I wanted to have at least one for demonstration. And here I just used traditional bitmap painting application and a drawing tablet. So I didn't work with a mouse here. But that's also personal choices, which tools you use for this markup. Another way is to work with simulations or combinations of real data and simulated data. And that is actually has been shown to be a pretty good way to extend your data. Can be done using geometric models, can be some kind of template models that you have a measured image, and on that measured image, which is, say, in high quality, you add noise of different levels. You maybe also add these spots that I was marking up. So that could be one way. And finally, you can also run physical simulations and use those to describe what you're doing. These are, of course, models. Sometimes they're doing pretty good. I've seen an example where people, actually, I think there is a link to it. Yeah, this Fuchs et al. They ran the whole system. They created a geometric model where they included some kind of inclusions or voids in a sample. Then they ran it through a Monte Carlo simulation to do the tomography. And afterwards, they used the machine learning to test. And then they had really the ground truth on the other side for this. So it appears to have resulted in very good results. Also, when they afterwards tested it towards human labeled data. So it's a way to proceed on also. And then there's the problems with all the data sets. One I have mentioned already is imbalance. You can have too few examples. Sometimes you only have one or two because the beam time didn't allow you to get more. Can also be that they are too homogeneous. There are not very sufficient many variations with it. And yeah, we are talking about experiments, so a lot of problems can come up. And there's, of course, when you do the analysis, that those problems, they propagate. And one problem is bias. And this was, I think it was, yeah, Google. Some of you probably already saw it. Google has a lot of picture labeling methods, and here they had pictures of skyscrapers, airplanes, cars, bikes, gorillas, and graduations. And in this case, it wasn't gorillas, and someone got really upset. It was two persons. And such problems can happen if you, for example, have used this ImageNet database where there's maybe a lot of gorillas in it, but maybe not so many black people. And well, that is what then happened in the training, that the configuration of these two and the skin color make this wrong decision. Google was quick to respond on this because this is really embarrassing. So what they did was just the quick fix. There are no gorillas in the database. So they can't find gorillas anymore. And then it worked. I think a better way to solve it is to actually increase the variability in the data. And here are some sample images from the IBM diverse face data set. And it's categorized not only with these categories, probably they also say more about it. So for example, wearing a hat, he actually also has glasses. So I believe each picture has a table of many, many, many features of these people. And with that variability, then it's better or easier to find, avoid this kind of biasing. So here is an example of data and labels. And here we have extracted only 1, 2, 3 from the MNIST database. And you can see in the example that we have a slight overrepresentation of 1s, and 2s and 3s, they are about the same. So here we can again see how the data is distributed within the data set. And limited data, there are different types of limited data. One is that you just have too few data points. Another one is you have unbalanced data. So you have one class that is really widely represented and one class that you barely see. If they're well separated, that's no problem. You can easily separate them from each other. But what happens if you have them overlapping like this? Then you only see a very weak bump on the main distribution, and it's very hard to distinguish. And the way to get around this is try to focus more on the underrepresented type, and hopefully getting better balance. So maybe do the training in a way that it sees more of the underrepresented type. Again, coming back to the roots and soil, you don't have to show all the soil. You can show some pieces of the soil. And of course, you have little or missing training data, that the training data is not completely labeled, but that can be worked out by do a lot of training yourself. So that leaves us to augmentation and a break. So let's have a short break. See you in 15.
I didn't see.
there is they are very much overlap.
Yeah.
Vocabulary is confusing in literature even.
Yeah.
So
Kind of like, the Venn diagram.
yeah, so kind of they are overlapping.
Yeah, I see it.
You sometimes you need, for example, segmentation to do detection.
Yeah.
So
Yeah.
it's a fuzzy world.
Yeah, but as long as I guess the end goal gets done, it doesn't really matter.